Hello everybody. In this video, I will be talking about the power of control in experiments. In other words, having a control group. I will be talking about two types of experiments, the ones that have control groups and those that surprisingly don't. And I will show you where it actually makes sense not to have control groups. And in the end, we will finish by covering the key challenges that we may encounter as researchers when we are trying to run an effective experiment with a really good control group. So let's jump right into it. Just as a refresher, experiment is a scientific method of investigation, which originated in natural sciences. That's why, as a refresher, I'm showing you an example from perhaps biology research where we are varying our dosages. And the dosage level is actually our independent variable. It's our cause, it's our intervention, and it is our x variable. The terminology may vary, so do not get confused. Different disciplines have their own particular ways of calling independent variable in experiment a particular name. But don't get confused because we are actually referring to exactly the same thing. What we are focusing on in this video is actually the control group, this one over here, and I will tell you what control group is and what it's not. An important aspect to keep in mind here is not to confuse the concept of a control group in the experiment, which is this one over here, with controlling for any extraneous variables. So what I'm referring to by extraneous variables, it could be any variable outside of your control which may potentially impact on your dependent variable when you measure it on its variations. In this very simple example, it could be, for example, time of the day, whether it's day or whether it's night, which may in fact impact on how this particular drug is absorbed in the body of an animal. I'm just hypothesizing, okay? It's just an example. But what I'm trying to highlight here is that controlling for any extraneous variables means that you, as a researcher, would like to isolate the impact of these extraneous variables on your experiment. In social sciences or in marketing or in psychology, that would mean that you would try to control for any potential individual consumer differences or individual differences in people, in your participants, after you executed your treatment and you measured the dependent variable. What am I referring to? It could be age, it could be gender, which may impact on differences that you're observing in social sciences experiments, or it could be some other psychological, psychographic variable. If, however, your psychographic variable forming part of your key investigation, it should form part of your hypothesis and it should act as a moderator because the moderator changes the relationship between your intervention and your dependent variable. Either makes this relationship stronger or it makes it weaker, in other words, annulling it. So this is an important distinction, and keep in mind, I do have a video on moderators on this channel, which is very detailed, and you can have a look at this video. So what do we actually need to accurately establish causality in our experiments? Well, we first need to manipulate our independent variable and have a group that is not exposed to this treatment, because we need to prove that exposure to this treatment leads to this particular change versus not being exposed to this treatment. It sort of makes sense. Then, ideally, it would be great to have a random allocation of the participants into your experimental conditions so that you rule out researchers' bias and any other bias. Then, what do you need to do? You measure your dependent variable after the treatment. And then you measure the differences in your dependent variable between the groups that you have in your experiment. So it's very basic, it's very simple, and it's very beautiful in its nature. Let's now have a look at experiments that actually have a control group. This is how I like to schematically visualize experiments that have control groups. So here, for educational purposes, it is in red color, and it is suggesting that when we have a control group, we are naturally comparing the difference between this control group and the other groups that receive different levels of your manipulation or the intervention, different type of an advertisement, different type of a video, different type of a newspaper article, different type of task, priming, framing, you name it. We are very creative in social sciences. But what it actually means is that in this type of a design, when you have control groups, you actually have to specify in the hypothesis what type of differences you are expecting between control group and the other groups, and also between the groups. So what do I mean by this? Visually, in terms of comparisons, in experiments that have a control group, we can accomplish comparison between the control group and all other groups. That's why I have versus here. It is very similar to intervention versus control. It's basically synonymous because it allows us to see what happens if we have this type of an advertisement versus the baseline. A person who, for example, sees a neutral advertisement or no advertisement whatsoever. I will talk about the types of control groups very soon. Here, as you can see, I have N, which means that you have any amount of groups in your experiment that receive treatment or intervention. But please do not get carried away with it because the more doesn't always mean the merrier or better when it comes to experimental research, because it may be challenging to statistically estimate the results, and it may be also challenging in terms of theory to formulate your hypothesis. So go back to the phenomenon that you study and have the number of groups that receive interventions meaningful for your research. Also, what's important to keep in mind here is that you need to formulate either a hypothesis of a difference between your control group and other treatments, or a hypothesis of no difference, where you would say, for example, there would be no difference between the control group and group one. Now, you may be thinking, what's the situation where that would be the case? Just wait for a while, and I will show you examples where it makes sense to have a hypothesis of no difference. And the ability to compare quantitatively the difference in your dependent variable in the outcome between the group is one of the unique aspects of experimental research where the causality can be established and the actual numerical difference between different types of intervention can be beautifully established. So overall, this is what's possible in the experimental research where we have a control group. An important point to keep in mind here is that because we are making multiple comparisons in such experiments, it's important to have sufficient sample size, and it's important to control for type 1 error, which is false positive. How do we accomplish this? When we have several groups, more than two groups, we can have an analysis of variance tests, and we can do post hoc tests, Tukey or Panfironi, depending on what you are trained in, depending on your discipline, to compare specific results between the groups. Multiple comparisons are very powerful because they can actually demonstrate between which groups differences exist and between which groups they actually don't exist. Now, let's have a look at the types of control group. Now, in theory of experimental research, there are two types of control groups. The first one is called active, and many of you may have heard of it under the term of a placebo group. And the second one is passive, which is termed as nocebo group. Try to remember it by thinking that there is nothing. No, nothing is there. And this is actually an example where your control group, your participants, do not receive any stimulus. But an active placebo group, as the name active suggests, means that they receive a stimulus, they receive a similar experience, but not on the expected level of variation that you have in your manipulations. And very often it has a nature of neutral stimulus, neutral information that participants are exposed to or they are receiving. Let me give you an example of a nocebo experiment. Let's stick with a very simple experiment where we have only two groups. The first group is a manipulation group where participants are receiving negative brand information. let's say information about product crisis. And after this information is received, we measure brand attitudes. The nocebo group, as the name no suggests, means they receive no information, no stimulus. And what happens usually here is that we just measure respondents' brand attitudes straight away. And the comparison that we are trying to make here is actually whether there is a difference in brand attitudes between the situations when people receive negative brand information, in other words, product crisis information, and instances when they don't receive any information about the brand. And let me show you an actual paper, real research, where a nocebo approach was used, and it made perfect sense to actually use a nocebo. This work was carried out by Wen, Zhang, and Zhang, and it was published in Journal of Business Research. And what happened is that researchers wanted to test several hypotheses, but here I have an example of the first hypothesis for you only. And what they were looking at, they were trying to see whether consumers have associations between the shape of food and whether this food is rated as healthy or less healthy. And they wanted to ultimately prove that consumers tend to associate angular shapes with healthier foods and circular shapes with unhealthier foods. They, as you can see, had four groups which they allocated randomly to different conditions, and they had a group that did not receive any treatment. So, in these four groups, in the first phase of their experiment, they allocated different tasks to each group. What did they do? They showed a carton of milk and sandwich to a group allocated to healthy angular condition, and they asked to rate it from 1 to 7, where 1 was extremely unhealthy and 7 was extremely healthy. Then they asked participants in the unhealthy angular condition to rate chocolate on the same scale. People allocated to healthy circular condition rated an egg and tomato in terms of its healthiness, and people allocated to unhealthy circular condition rated cola and hamburger on the same scale. People in the nocebo group did not do any of these rating tasks. And in the second phase of the research, they exposed them to a target stimuli of four different foods, which was circular healthy, circular unhealthy, angular healthy, and angular unhealthy. And they also had a distractor stimuli, which I'm not showing here. And these are not the actual images used in the journal article. If you're interested to see what they use, please go and check this reference here, available at the bottom of the page. What did they actually find? They found, as predicted, no difference between healthy angular, healthy circular conditions, and the control group. And this is exactly what they wanted to prove. They wanted to prove that consumers inherently, whether they're provided with some stimulus or they're not provided with any stimulus, have specific, unique associations regarding the shape of the food and whether they associated with healthy or less healthy foods. So the nocebo group here is used beautifully because it helps to establish that inherent learned associations that we have. Now, let me show you an example of a placebo group. I'm using exactly same example, very simple one with two groups, where the first one again is receiving negative brand information about product crisis. But in this case, when we have active placebo group, they receive a treatment. And because it needs to be approximated to the experience in the manipulated condition, they need to receive brand information. But in this case, we do not make it negatively charged, we make it neutral. For example, we may say that a company is planning to extend its product line. And we need to hope that this sort of information will be perceived neutrally by our participants. But that's why we do pilot tests as well. What happens after the exposure to one of these conditions? Participants rate a brand and we measure their attitudes. And in this case, the comparison is different. It is Are there any differences between receiving negative information about the brand and receiving neutral information? And that is different from the nocebo group, which means receiving no information whatsoever. Now, you may be wondering which one is more recommended in the literature. Is it the active placebo or is it nocebo? The literature says that it's better to have an active placebo, but Woodside, in the work published in 2016, also recommends to use active placebo group and passive nocebo group when unsure, because in this case, you would be able to see are there any differences in your dependent variable depending on whether you receive some sort of information, but it's neutral, versus you don't receive any information, versus you receive a treatment. So please, again, have a look at your experiments. What are you trying to accomplish? What phenomena you study? And which theory you're using in order to make the right choice about the control group and its type for your experiment. Now I will show you an example of a paper which used active placebo and passive nocebo group, but they didn't use it at the same time, but in different experiments that they ran. And you will see how each of them serves to accomplish a particular goal and prove a point, prove hypothesis. This work comes from White and Simpson's work published in Journal of Marketing in 2013. Over here on the screen, I have the interventions, the manipulations that they executed, and they had an active placebo group in which they showed information about particular behavior. This was their first study. It was a lab online experiment. It had random allocation of participants to the groups. And in the end, they were trying to measure grass cycling attitudes. And the overall purpose of this research was to see whether injunctive versus descriptive norms and also the appeal that you use, whether it's individualistic or collective, impact perceptions of graph cycling. Injunctive norms basically are about you should do something. Descriptive norms is people are doing something or this behavior. Individual appeal is basically saying your neighbors want you to grass cycle, and collective appeal is basically saying our neighbors want us to grass cycle. So as you can see, they had the complex design and different combinations and variations of social norms that they used and also the appeals. And then as an active placebo group, they had information only about the behavior about grass cycling. So, what they ultimately wanted to achieve here is to see which of those appeals results in more favorable attitudes about grass cycling as one of the sustainable behaviors, and how is it different from information only. Then I will show you their second study, which was a field experiment where they distributed leaflets to 676 households. Here I have only two examples, they had more. But what they were trying to do in this case is they had groups that received the treatment, different types of information, but they also had a group which did not receive any treatment, and this served as their control condition. What did they do? They measured the number of bags of garbage and the amount of grass in the bins before they administered the interventions and some time after. And they made comparisons between these groups and this particular neighborhood or suburb that they picked, which did not receive any treatment whatsoever. And this was done to see what is the difference between these type of messages versus not receiving any message whatsoever. So as you can see here, the purpose is slightly different, and the passive nocebo group beautifully serves this point. The only thing to keep in mind is that when you run designs like this with field experiments where we naturally have less control, we need to ensure that we control for any extraneous variables. For example, we need to ask all of our participants, because we have less control over the experimental procedures, whether during this time frame they received any information about grass cycling or any other sustainable knowledge information that can potentially impact on the outcome at time 2. So this is very important to keep in mind because ultimately you want to establish that it is your intervention that is driving that change, that is explaining those differences in the dependent variable in consumers' behavior. Let's have a look now at experiments that actually don't have a control group. And in this case, as the name suggests, we don't have any group that receives no treatment or is not exposed to a sort of stimuli. The comparison here is taking place just between our intervention groups. And again, if you have more than two groups, it's important that you run post hoc analysis, multiple comparisons. You can use analysis of variance to actually try to reduce the type 1 error and establish between which of those groups actually statistically significant differences exist. In other words, differences that are not due to chance. Let me now show you an example of an experiment where it makes perfect sense to have no control group whatsoever. This research comes from Ni and Juan's work, which they published in 2023 in the Journal of Consumer Behavior. And in this type of research, they were trying to see whether there is difference in perceived effort and subsequent service evaluation depending on who delivered the service. And in the first group, they had a humanoid service robot in a restaurant and they compared to a human chef in a restaurant. So, as you can imagine, in this particular case, which is about service delivery, it's impossible to have a control group which could have a neutral message because you don't know who to put there, a robot and then a neutral message, or a chef and then a neutral message. It doesn't make any sense. And it makes no sense here to have a group that does not receive any experience because when you go to a restaurant and your research is about service delivery, someone or something has to deliver that service. So if you have a control group that does not have any treatment, it makes no sense and it's not helping you to find answers to your question. So I hope now you can see that experiments without control groups are possible and they do make perfect sense. It all depends on the research context and the phenomenon that you study. Let's now talk about challenges of designing effective control groups. There is a number. Let's start with the first challenge, and it is consumer expectations affect no treatment. In other words, our control group. What am I talking about? Sometimes consumers may have certain expectations that they will be taking part in the experiment. In other words, they will be receiving some exciting information. So that may affect how people allocated to a nocebo control group or a placebo control group will react to your dependent variable when you're actually measuring this. Why would that be the case? Because very often, due to ethics requirements, we have to give a participant information about the purpose of the research. Or we may say that you will be viewing a newspaper article or you will be reading something about a firm and then pay attention to it because we will be asking you questions about this firm. So participants, when they read the purpose of research, the introductory page to your experiment, they may just get a bit overexcited, which means that when you allocate them to a very neutral condition, they may still rate your brand quite favorably. And that would mean that you have very strange results in your control group versus your treatment. Try to keep your introduction very general. Don't overexcite them. Have several distractors so it's hard for them to guess the real purpose of research so that they answer in a more natural to them, not overexcited manner. Challenge number two. Sometimes it is very challenging to create a control group that would mimic the marketing experience, the decision making process that consumers naturally experience without producing any effects on them. I will give you an example. Sometimes it may be challenging to design a very effective control group and create a neutral message when we have to expose consumers to sensory information, to symbolic cues. They have to view particular packaging. You are measuring storytelling or you're looking at branding. It may be hard to create a neutral experience when we test the effects of music on consumers because There's a lot of variations how people perceive music. It may be challenging to recreate experimental design for the control group in such a way that you would create a genuinely neutral experience if we are talking about active placebo group. It may be also challenging to have a control group that does not receive any treatment if you have to naturally expose them to some sensory visual stimuli, to text, to a video. So you really need to think a lot about your experiment and identify whether you can effectively design a control group in a way that it's neutral and it will not really contaminate the results, will not produce strange results in your control group versus your manipulation groups that you are studying. Another challenge of control groups is a Hawthorne effect. And it applies to situations when consumers purposefully adjust their responses when they know that they are being studied. In other words, they change their behavior and they respond in a fashion that is very different from their natural decision making if they were not taking part in your research. I'll give you an example. Very often that takes place with research that has some ethical implications. We're trying to measure ethical and unethical behavior, political voting, or behaviors that may trigger responses towards the socially desirable dimension, such as sustainability, where, for example, you're trying to research how you can improve sustainable behaviors of consumers, and participants understand that they take part in research, they figure out the purpose. They may not necessarily want to purchase that more expensive product that you're testing in your experiment, but they change their behavior and say, of course, yes, I will. And you have very inflated high ratings in your interventions and in your control group because they are trying to provide socially desirable answers. In this case, the trends that you're observing in your experiment, they are not the true trends because participants are lying to you because they adjusted their responses and behavior. Because they know they are being watched, they're taking part in research. So, what some researchers are doing, they integrate a question in their experiments asking, did you genuinely respond to my questions at the end of the survey? Then, if the participant, at this case, genuinely told us no, then we screen them out. But this is a really tricky one. This is really a challenging one. Non-random assignment to the control groups and other groups in your experiment may also lead to big problems because in this case it introduces researchers bias. In this case, the pre-existing differences between your participants may be driving difference in the dependent variable, not necessarily your treatment, not necessarily those amazing hypotheses that you have designed and those stimuli that you have designed. And in this case, the undesirable outcome is that outcomes they differ between the groups, but for the reasons which are not related to your manipulation, to your intervention. So you are making incorrect causal inferences from the data, which is not ideal, really. So often, or I should say always, 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 try to have a random allocation of participants to your group. Use random number generator or use quadratic randomizer or randomizer in any other online software that you're using for your online experiments. Next challenge, individual psychological differences are really hard to control for. And we are also different. We are not just oranges and apples, we are limes, we are mandarins, tangerines, lemons, and I think I've covered it all, but it shows you that just like fruit can be very diverse, so are humans, if not more. What am I referring to? I'm referring to instances when differences in your control group and interventions may be due not to your interventions, but due to inherent individual differences, how they may differ in information processing, in involvement while reading your stimuli, while watching your video. They may vary in skepticism towards advertising, skepticism towards research. They may have their own expectations. They may have different personal values. They may have different knowledge. They may have different experience, and so on and so forth. And the danger here again is that the outcomes we observe in the groups, they may be different. The control group may be not different from other groups, and that will not align with your hypothesis, or there could be higher ratings or lower ratings than you expect. In this case, my recommendation would be if those individual differences based on theory, based on past readership, past studies, can impact on information processes, do include them as your moderators. Make them form part of your hypothesis because in this case, you not only control for them, you actually actively studying them and trying to understand under which condition the message will not get through, for example, or something else would happen with that stimuli that you are sending to your participants. If they form part of your moderators, those individual consumer differences, always measure them before your manipulation. This is what the literature is telling us. Now, this was everything for the control groups, their types, experiments with control groups or without them, and also the challenges that we may confront when we run experiments. I hope you feel empowered now, you know what to look out for, and you know how to design your control groups. Thank you for watching, and I will see you in the next one.